The next organ system that we are looking at in systemic anatomy and physiology is the cardiovascular system. The cardiovascular system is made up of the heart, the blood vessels that enter and exit the heart, as well as the blood that is contained within those blood vessels. For the anatomy of the cardiovascular system, we will cover the structure and function of the cardiovascular system, the location and structure of the heart, the different types of blood vessels, the various components that make up blood, and the different blood types. So getting straight into it, learning objective number 1, to describe the structure and function of the cardiovascular system. So as I mentioned, the cardiovascular system is made up of the heart, the blood vessels, and the blood. Cardia refers to heart, and vascular refers to the blood vessels in which the blood is transported. Now, the major function of the cardiovascular system is transportation. So the cardiovascular system works really closely with the respiratory system, as we learned about in the previous series of lectures. It carries oxygen-rich blood to the tissue cells, as well as deoxygenated blood back to the lungs. Blood transports nutrients from our gastrointestinal tract to our body tissues, and waste products to various organs to be expelled. The cardiovascular system is also really important for the endocrine system, with blood transporting hormones to their target cells. The cardiovascular system also plays a role in regulating a number of our body fluids. For example, blood helps regulate pH through the use of buffers like bicarbonate. It also helps adjust our body temperature through the redirection of blood to different areas of the body, as well as the proximity of blood vessels to the surface of the skin. Another important function of the cardiovascular system is protection, with blood protecting against excessive blood loss after an injury in the way that it clots, white blood cells protecting against disease by undertaking phagocytosis, and the presence of antibodies in the blood playing a role in our immune response. So the cardiovascular system is a big team player in the way that it interacts with all the other body systems, as well as a really important contributor to maintaining homeostasis. So now that we know what the cardiovascular system does, the rest of this lecture is going to focus on the anatomy of each of the components that make up this system. So starting with the heart, learning objective number 2 is to describe the location and structure of the heart. So the heart lies in an anatomical region that we call the mediastinum. The name mediastinum comes from a Latin word which just means middle. The mediastinum is this region outlined in a black dashed line here, and it encompasses the heart as well as the major corresponding blood vessels. The apex of the heart is this pointed tip. It's primarily made up of the left ventricle, and the apex of the heart actually rests on our diaphragm, which remember sits underneath our lungs here. The base of the heart is the upper flatter portion of the heart. It's formed by our left and our right atria. And despite how important the heart is, it is only a relatively small organ, about the size of your closed fist. So about 300 grams in adult males and 250 grams in adult females. Surrounding the heart, we have a pericardium. So you can see it on this image here on the left, surrounding the outside of the heart. You can also see it in a bit more detail on this image on the right. And remember, peri means around, and cardia refers to the heart. So the pericardium is actually made up of 2 main parts. We have the fibrous pericardium. That's this portion here. It's a tough, inelastic outer layer that prevents the heart from overstretching, as well as anchors the heart in the mediastinum while also allowing for the movement associated with the heart beating. The deeper serous pericardium is a thinner, more delicate membrane that forms a double layer around the heart. It has an outer parietal layer, which is this like white line here. That's actually fused to the fibrous pericardium, We also have an inner visceral layer, which actually makes up part of the heart wall, and therefore it adheres tightly to the heart itself. Between those two layers of the serous pericardium, we have the pericardial cavity. This secretes a pericardial fluid, which reduces the friction between these two layers as the heart contracts and as it moves. So recapping all of that in words for you, the pericardium is a membrane that surrounds and protects the heart. It's made up of two main parts: the superficial fibrous pericardium, which is tough and inelastic and prevents overstretching, while also anchoring the heart to its spot in the mediastinum. We then have the serous pericardium, which is the thinner inner membrane, which creates that double layer around the heart. This double layer is made up of our outer parietal layer, which is fused to our fibrous pericardium, and our inner visceral layer, also called the epicardium, which is stuck to the heart itself. In the space between these two layers of the serous pericardium, we have the pericardial cavity. This secretes our pericardial fluid, which reduces the friction between these two layers as the heart beats. Now looking at this same image again, but we're now going to focus on the wall of the heart. So the wall of the heart consists of three layers. So there's lots of layers in this small area here. We have the epicardium, which is the outermost layer, and remember epi means on top. We have the myocardium, which is this thick middle layer here, and myo refers to muscle. And then we have our endocardium, which is the innermost layer, with endo meaning within. So the epicardium, as we just mentioned, is the visceral layer of the serous pericardium. It provides a smooth, slippery texture to the outermost surface of the heart, as well as contains blood vessels and lymph tissue that supply the underlying myocardium. The myocardium is our middle layer of the heart wall. It's responsible for the pumping action of the heart, with the cardiac muscle contained within this section involuntary, meaning that they contract without us needing to think about it or put any conscious effort into it. The innermost layer is our endocardium. The endocardium provides a smooth lining for the chambers of the heart. It also covers the valves of the heart and the inner lining of the main blood vessels that enter and exit the heart. The smooth lining of the endothelium minimizes the surface friction as blood passes through the heart, so it can move through really easily. Again, putting all of that into words for you. So the outer layer of the heart is our epicardium. This is made up of the visceral layer of the pericardium, which remember is that double membrane that surrounds the heart. The thick inner layer is the myocardium, which is made up of the involuntary cardiac muscles. The innermost layer is our endocardium, which is continuous with the valves of the heart and the major blood vessels, and is smooth to reduce the friction experienced as the blood flows through the heart. Now taking a look inside the heart, and the heart is made up of four chambers. There are two chambers called the atria, so these two superior chambers here, and then two inferior chambers called the ventricles. The two superior atria receive blood from the vena cava and the pulmonary veins. The two inferior ventricles eject blood out into the pulmonary trunk and into the aorta. Now we're going to look at these four chambers in more detail in a second, but there's two structures that I just wanted to show you on the anterior surface of the heart that relate to the function of the atria. And these are these two pouches here and here, and are called auricles. So the auricles of the heart sit on top of the atria, and they act a little bit like a pocket. So when blood is filling into the atria and there's a bit of an overflow of blood, it can go into these pockets so that the atria can actually hold more blood volume. But coming back to the internal chambers of the heart and starting with our right atria here. So the right atria forms the right superior surface of the heart, and it receives blood from 3 veins. We have the superior vena cava, the inferior vena cava, and our coronary sinus. Now you can't actually see the coronary sinus in this view here. We can see the opening up into the right atria here. And the coronary sinus is actually the vein that drains the blood from the heart tissue back to the heart. The wall of the right atria is about 2 to 3 millimeters thick. And in between our 2 atria, we have a septum or a membrane called the interatrial septum. Blood passes from our right atria through a tricuspid valve into our right ventricle. The tricuspid valve, which we will look at in more detail shortly, is called the tricuspid valve because it has 3 cusps or 3 flaps. This tricuspid valve can also be called the right atrioventricular valve because it sits between our right atria and our right ventricle. Now moving on to our right ventricle, and this is our right ventricle here. It makes up most of the anterior surface of the heart. The walls of the right ventricle are a little bit thicker, about 4 to 5 millimeters. And internally, between our right and our left ventricle, we have our interventricular septum. As I mentioned on the last image, as blood moves from our right atria into the right ventricle, it passes through an open tricuspid valve. The cusps or the flaps of our tricuspid valves are actually connected to tendon-like strings that we call the chordae tendineae, which are in turn connected to papillary muscles, which anchor them down into this right ventricle. From the right ventricle, the blood then passes through an open pulmonary valve. This can also be called a semilunar valve. It goes into a large artery called our pulmonary trunk, where it splits into our right and our left pulmonary arteries to go to our right and left lungs. After returning from the lungs, the blood then enters our left atrium. This is our left atrium here. It's about the same thickness as our right atrium, and it forms most of the base of the heart. The blood that enters our right atrium does so through our 4, so 1, 2, 3, 4 pulmonary veins. Blood then passes from our left atrium through an open bicuspid valve, because we only have 2 flaps or cusps in this valve, down into our left ventricle. Again, we're going to come back to our bicuspid valve, but you might also see this valve termed the mitral valve or the left atrioventricular valve. Lastly, the blood moves from the left atria into our left ventricle and then out around the body. The left ventricle makes up a large portion of the apex of the heart, and it has the thickest walls of all of the chambers, averaging 10 to 15 millimeters. Like the right ventricle, we have chordae tendineae that anchor the bicuspid valves or the flaps of the bicuspid valve to our papillary muscles and then the left ventricle. When the ventricle contracts and blood passes up out to the aorta, it passes through an open aortic valve or an open semilunar valve. The blood then goes into the aorta. Some will branch off into coronary arteries, which are the small arteries that feed the heart muscle. The rest will go to the branches of the aorta or the descending aorta and carry blood through the rest of the body. So to summarise the movement of blood through the heart, and this is also a really good revision for trying to identify all of the structures. Blood enters into the right atrium through our superior and inferior vena cava, as well as our coronary sinus. The blood goes from our right atria through an open tricuspid valve into our right ventricle. From our right ventricle, the blood goes up through our pulmonary or our semilunar valve into our pulmonary trunk. That splits off to go into our right and our left pulmonary arteries and to the lungs to become oxygenated. Returning from the lungs back to the heart, we have the blood entering through our 4 pulmonary veins. That enters into our left atrium. The blood moves through an open bicuspid valve into the left ventricle. When the left ventricle contracts, it pushes blood through an open aortic or semilunar valve into the aorta. These branches of the aorta and the descending aorta take blood to the rest of the body. You can't see it here, but we also have smaller vessels that branch off and go back and feed this heart tissue itself. Now, while we're on this image, just one other thing that I wanted to point out to you. So we have our tricuspid and bicuspid valve. A little tip from remembering which is which that I got told was that you try before you buy. So your tricuspid valve and your bicuspid valve. So just focusing a little bit more on the valves of the heart and how they function before we move on to some other structures. And so as each chamber of the heart contracts, it pushes blood into a ventricle or out of the heart into an artery. Valves open and close in response to pressure changes as the 4 chambers contract and relax. The function of the 4 valves is to ensure that the blood flows in one direction, and they do this by preventing the backflow of blood. So the tricuspid and bicuspid valves, also termed the atrioventricular valves because they sit between the atria and the ventricle, when they are open, their cusps hang into the ventricles. This allows blood to move from the atria into the ventricles. When they are open, the papillary muscles are relaxed, the chordae tendineae are slack, and we have this opening between the 2 chambers. When the ventricles contract, the pressure in this chamber pushes those cusps upwards where they will meet, losing this opening between the 2 chambers. When this happens, our papillary muscles also contract. It pulls our chordae tendineae tight, so that despite the blood pushing up this way into those cusps, they won't avert into the atrium, so that we have no backflow of blood. The blood goes only out the pulmonary trunk or the aorta in the direction that we want it. Looking at our semilunar valves, and they work in a slightly different way. Our semilunar or our aortic and pulmonary valves are termed semilunar because they're made up of 3 crescent moon-shaped cusps or flaps. Each of those cusps or flaps is attached to the corresponding arterial wall, so either our pulmonary trunk or the aorta. Now, the semilunar valves work by allowing the ejection from blood from the ventricles into the arteries, but preventing the backflow of blood back into the ventricles when they relax. So if we say this ventricle here contracts, it forces the blood up through this open, it opens up this this pulmonary or semilunar valve. When this ventricle then relaxes, this pressure within the ventricle drops, some of that blood is going to want to trickle back into the ventricles. How these flaps of the semilunar valves work is that they will then capture this blood that's trickling back in the opposite direction. When it fills up with blood, the edges meet and it blocks off this pathway, so the blood can only move in that one direction, and they will only reopen again when this ventricle contracts and pushes more blood out through that pulmonary trunk. I kind of visualize it as an upside down umbrella that's catching all of the rain. So once the blood circulates around the heart and the lungs, it goes back to the heart before travelling around the body in our blood vessels. So learning objective number 3 is to list and describe the different types of blood vessels. But before we get to the actual different types of blood vessels, I really quickly want to walk you through the basic structure of a blood vessel. So the wall of a blood vessel, just for something new, consists of 3 layers, which we also call tunics. We have an epithelial lining, a middle layer of smooth muscle and elastic connective tissue, and a connective tissue outer covering. So the 3 layers of a blood vessel from inner to outer are our tunica interna, our tunica media, and our tunica externa. So the tunica externa forms the innermost lining of a blood vessel, and it's what's in direct contact with the blood. Itself is made up of 3 layers. We have an inner layer of endothelial cells, which is continuous with that lining of the heart. It's nice and smooth so that the blood can flow really easily through these vessels without generating too much friction. Underneath our endothelium is our basement membrane. This provides physical support for the endothelium. The outermost layer is our internal elastic lamina. This has several holes, as you can see through here, and this allows the diffusion of substances through that tunica interna through to our thicker tunica media. Next, we have our tunica media, so this is that middle layer, and this is where we see the greatest variation between the different blood vessel types. In most vessels, it's a relatively thick layer comprising of mainly smooth muscle cells and elastic fibres. The primary role of these smooth muscle cells, which extend in a circle kind of around the blood vessel, is to regulate the diameter of the lumen or the opening of the blood vessel. So vasoconstriction is when the smooth muscle contracts and that diameter of the blood vessel gets smaller. Vasodilation is when those muscles relax and the diameter of that lumen increases, so the vessel gets bigger. 2 important terms to know. In addition to regulating blood flow and blood pressure, this smooth muscle layer also contracts in a small artery or arterial when it's damaged to help limit the loss of blood through an injured vessel. This process is called vascular spasm. The tunica externa is the outer covering of a blood vessel and consists of elastic and collagen fibres. The tunica externa contains numerous nerves as well as tiny blood vessels that supply the tissue of the vessel wall, particularly the large blood vessels like the aorta or the pulmonary trunk. This layer also helps anchor the vessels to the surrounding tissues so it doesn't get damaged when we move around. So moving on now to the different types of blood vessels, and there are 5 main types. So starting from this side of the slide here, we have our arteries, arterioles, capillaries, venules, and veins. Arteries carry blood away from the heart. The large elastic arteries that exit the heart then branch into smaller arteries to take blood to different regions of the body. These arteries then continue to divide into smaller arteries until we call them arterioles. Arterioles enter our tissues and then they branch into several smaller vessels, which we call capillaries. The capillaries have a very thin wall, which allows the exchange of substances from the blood into the body tissues and then from the body tissues into the blood. A group of capillaries will then reunite to form a venule. These in turn then merge to form progressively larger blood vessels, which we call veins. Veins are the blood vessels that conduct blood from the tissues back into the heart. So from biggest, we've got artery, arteriole getting smaller, and then the smallest is our capillaries, and then getting larger again, we have capillaries to our venules to our veins. Now, a little bit of text information to provide some context for each of those different types of vessels. So starting with arteries, our arteries carry blood away from the heart. And like all blood vessels, the wall of an artery has 3 layers. However, one distinguishing feature of an artery is that it has a thick muscular and elastic tunica media, or that middle layer. The high proportion of smooth muscle fibers in this layer makes these vessels capable of dramatic vasoconstriction or vasodilation, so increasing the diameter or decreasing the diameter. And this allows them to adjust the rate of blood flow. The high proportion of elastic fibers also gives these arteries high compliance, meaning that their walls stretch easily or they expand easily without tearing in response to an increase in blood volume and blood pressure. So like when the heart ejects all the blood out of the left ventricle. And within the category of arteries, we actually have two separate categories or two separate types of arteries. We have elastic arteries and we have muscular arteries. Our elastic arteries, which are the largest arteries in the body, range in size from the size of a garden hose, like in the aorta and the pulmonary trunk, to finger-sized branches that come off the aorta. And despite these arteries having the largest diameter, elastic arteries actually have quite thin walls compared to the overall size of the vessel. These arteries help move the blood forward while the ventricles are relaxing. As blood is ejected from the heart into these elastic arteries, their walls will stretch, so easily accommodating that big influx of blood. When the ventricles relax, those elastic fibers will then recoil or get smaller again, and that helps push the blood forward. The other type of artery is our muscular arteries. In these arteries, there is more smooth muscle and less elastic fibers. These arteries can regulate the flow of blood to various organs due to this thick muscular layer. And they range in size from the diameter of a pencil to as little as 0.5 millimeters in diameter. Moving further away from our heart, and our next type of blood vessel is an arteriole. Arterioles are abundant microscopic vessels that regulate the flow of blood into our capillary networks, which we find within our tissues. We have approximately 400 million arterioles, and they range in size from 15 to 300 micrometers in diameter. The tunica externa, or the outer layer of an arteriole, consists of areolar connective tissue with many sympathetic nerve fibers. This nerve supply can alter the diameter of the arterioles and thus alter the rate of blood flow and resistance through these vessels. So how quickly the blood gets to those capillary networks. When the diameter of a blood vessel is smaller, friction is greater, and therefore there will be more resistance to blood flow, and it will flow slower. Capillaries are then the smallest blood vessels with diameters of between 5 and 10 micrometers. As an example of how small our capillaries are, red blood cells have a diameter of 8 micrometers, and therefore they must actually fold on themselves to pass single file through the lumen of our capillaries. Capillaries form a U-turn of blood vessels which connect the blood exiting the heart in our arterial system and the blood returning the heart via the venous system. And they form an extensive network, approximately 20 billion in number, of short branched interconnecting vessels that course along our individual cells of the body. This network of capillaries forms a big surface area to make contact with the body cells. The primary function of our capillaries is the exchange of substances between the blood and the interstitial fluid. Think of the capillaries that surround the alveoli. Capillaries are therefore found near almost every cell in the body, but their number will vary with the metabolic activity of the tissue that they serve. So tissues with high metabolic requirements, like your muscles, the brain, the kidneys, will use more oxygen and therefore have extensive capillary networks. Tissues with lower metabolic demands, such as our tendons and our ligaments, will contain fewer capillaries. Capillaries are absent in a few tissues, so the cornea and the lens in the eye, as well as the cartilage that surrounds many of our bones. The structure of our capillaries is well suited to their function as exchange vessels because they lack a tunica media and a tunica externa. So they actually only have that inner layer of the blood vessel wall. And because it's only one single layer of endothelial cells and a basement membrane, substances in the blood can pass really easily through this one layer of cells to reach the blood or to go from the tissues back into the blood. Having now completed a U-turn, we're going to talk about the venous system, which is responsible for bringing the blood back to the heart. So veins, unlike their arterial counterparts, have quite thin walls that do not readily maintain their shape. Venules drain blood from the capillaries and begin that return of blood back towards the heart. The smallest venules measure between 10 and 50 micrometers in diameter and are quite porous. So there can be a little bit of exchange and diffusion between our venules and body tissues. As they move back towards the heart, the diameter increases to about 50 to 200 micrometers. They also have thicker, more muscular walls. And so in these muscular venules, we can't have that exchange or diffusion of substances. The last type of blood vessel is our veins. Veins are thin-walled and much smaller compared to our arteries. They range in size from 0.5 millimetre up to about 3 centimetres in diameter. They're composed of essentially the same 3 layers as our arteries, but the relative thickness of each of the layers is different. So the tunica interna, or the inner layer of the veins, is much thinner than that of the arteries, as well as the tunica medica, because there's much less smooth muscle and elastic fibres. The tunica externa, or the outer layer, is the thickest layer of our veins, and it consists of lots of collagen and lots of elastic fibres. Now, the lumen, or the opening of a vein, is bigger than an artery, despite the vein itself being actually smaller. This is due to the thinner layers, that surround that vessel. When blood is not running through a vein, so like in the cadavers that we have in the lab, the vein will often appear flat or collapsed. Now, many veins, especially those in the lower limbs, also contain valves. They're thin folds of that tunica interna that form flap-like cusps. The low blood pressure in veins means that the flow of blood returning to the heart can actually slow and sometimes even back up. The valves aid in venous return, or the blood going back to the heart, by preventing too much of this backflow occurring. Moving on now to what we find within our blood vessels, and that is blood. So learning objective number 4, to list and describe the components of blood. So a little bit about blood in general, and blood is denser, thicker and stickier than water. It sits at a temperature of about 38 degrees Celsius, which is approximately 1 degree hotter than what you'll see for your oral or your rectal body temperature. It has a slightly alkaline pH, ranging between 7.35 to 7.45. The colour of blood varies with its oxygen content. When blood is highly saturated with oxygen, it is bright red. When it is less saturated, or as there's less oxygen bound to the hemoglobin in the red blood cells, the blood appears darker. The total volume of blood is between 5 and 6 litres in an average size male and 4 and 5 litres in an average size female. The gender difference between blood volumes is purely due to body size. Now, blood has 2 whole components: blood plasma, which is a watery liquid extracellular matrix that contains dissolved substances and makes up about 55% of total blood volume. And then formed elements, which are the cells and the fragments, which make up the remaining 45%. In a sample of blood, if it's centrifuged or spun in a small glass tube, the cells, which are more thick, will sink to the bottom, while the plasma will form that top layer. As I mentioned, about 45% of our whole blood is formed elements. Normally, of this 45%, 99% will be red blood cells, with white blood cells and platelets making up less than 1%. And this is what we term the buffy coat that you see in between those two different parts. So looking at each of those components of blood individually, and when the formed elements are removed from the blood, a straw-coloured liquid called blood plasma is what's left behind. So blood plasma, or you can just call it plasma, is about 91.5% water, 8.5% solutes, most of which are proteins. These proteins include albumins, globulins, fibrinogens, and then a small number of other proteins. Specific examples of proteins within blood plasma include antibodies, which are produced during certain immune responses. Foreign substances such as bacteria and viruses can also stimulate the production of millions of different antibodies. An antibody will bind to the antigen or the protein that actually stimulates its production, and that's how it disables that invading antigen. Besides our proteins, other solutes in plasma can include electrolytes, nutrients, gases, regulatory substances, so things like our enzymes and our hormones, as well as waste products. Moving on to our formed elements, and these include our white blood cells, our red blood cells, and our platelets. Our red blood cells, or erythrocytes, transport oxygen from the lungs to the body cells and deliver carbon dioxide from the body cells to the lungs. White blood cells, or leukocytes, protect the body from invading pathogens or foreign substances, and there are several types of white blood cells, including neutrophils, lymphocytes, monocytes, eosinophils, and basophils. Platelets are then fragments of large cells that do not have a nucleus. Among a few other actions, they release chemicals that actually promote blood clotting when blood vessels are damaged. So taking a bit of a closer look at each of those formed elements, and red blood cells make up most of this component of blood. So red blood cells are bioconcave discs, which is this shape here. Kind of looks a bit like a fruit tingle. They have a diameter of about 7 to 8 micrometers, which equates to about 1/1000th of a millimeter. So very, very, very small. Red blood cells are strong, but quite flexible, which allows them to bend but not rupture as they squeeze through our really narrow blood capillaries. Our blood cells actually lack a nucleus and organelles, which in some ways is good because they don't have these internal, organs. All of their internal space is available for oxygen transport. Within the cytosol of our red blood cells is hemoglobin, which we mentioned briefly when we looked at the respiratory system. Each red blood cell contains about 280 million hemoglobin molecules. So hemoglobin molecules consist of globin, which is a protein made up of 4 polypeptide chains, and a ring-like pigment called heme or heme. To each heme is an iron atom, and it's here that the oxygen actually binds to the hemoglobin. So we have our 4 chains of polypeptides here. We have our heme is the yellow, the red circle is our iron, and that's where the oxygen molecules will bind to this hemoglobin. White blood cells, also called leukocytes, are different from red blood cells in that they do contain a nuclei as well as a full complement of our organelles. They are far less in number compared to red blood cells, although they do increase in number as a protective response to things like stress, invading microbes, strenuous exercise, and surgery. Once a pathogen enters the body, the general function of the white blood cells is to combat them by phagocytosis or another type of immune response. To accomplish some of these tasks, many blood cells can actually leave the bloodstream and collect at sites to fight injury and infection. Elevated levels of white blood cells can indicate things like infection. Low white blood cell counts can be indicative of diseases like leukemia. Now, white blood cells are classified as either granular or agranular, depending on whether they contain or don't contain chemical-filled cytoplasmic granules or vesicles that you can see when you stain the cell and look at it under a microscope. Our granular leukocytes include our neutrophils, eosinophils, and basophils, and you can see those granules in the darker bits there. Our agranular leukocytes are our lymphocytes and our monocytes. Now, we don't actually cover the immune system in systemic anatomy and physiology, but I know that this will be of interest to many of you, as well as something that many of you go on to study in more detail in future units. For that reason, I have included this slide here that has each of the different types of the white blood cells, as well as a bit of a function on how they work. I'm not going to go over this now, and this is not something you will be tested on, but I thought I would just provide this information for you if you would like to take a look. Our platelets are then the final formed element that makes up blood. Platelets are fragments or pieces of giant cells called megakaryocytes. Because it is a fragment or a smaller piece of a bigger cell, each is an irregular disc shaped, about 2 to 4 micrometers in diameter. Platelets also contain no nucleus, and their primary function is to promote blood clotting. Granules or vesicles within the platelets, once released, promote blood clotting through chemical messaging. Platelets help stop blood loss from a damaged blood vessel by forming a platelet plug. And you can see an example of a platelet plug here. On the topic of blood, our final learning objective for anatomy of the cardiovascular system is to describe the different blood types. And when I say blood types, I'm talking about your blood group. So on the surface of red blood cells, there are an assortment of antigens, which are little extensions that stick out of the plasma membrane, made up of glycoproteins and glycolipids. Blood is characterized into different blood groups based on the presence or absence of these antigens, and there are consequences of these different blood groups, particularly when it comes to things like blood transfusions. Now, there are actually lots of different antigens that stick out of the plasma membrane on a red blood cell, and your blood can be classified in a number of ways, but there are typically 2 main types that we will generally look at. These are our ABO blood groups and our RH or our rhesus blood group. So our ABO blood group is based on the presence or absence of 2 different antigens. We have antigen A and antigen B. People whose red blood cells display antigen A only will have type A blood. Those who have Those who have antigen B only have type B blood. Individuals who have both antigen A and antigen B antigens will be type AB blood, and those that have neither are type O blood. So if you have A antigens, type A, B antigens, type B, both antigens, type AB, and then neither antigens, type O. Now, where this blood type gets important is that in our blood plasma contains antibodies which react with the A and B antigens. So we have anti-A antibodies that react with antigen A and anti-B antibodies which react with antigen B. Now, when a red blood cell comes into contact with a specific antibody, that antibody binds to the antigen and it causes clumping or agglutination, so AGGLUTINATION. When the antigen and the antibodies combine or bind, they then activate an immune response, which can cause the rupture of our red blood cells. So if you are blood type A, you will have anti-B antibodies. If you're blood type B, you will have anti-A antibodies. If you're blood type AB, you will contain neither antibodies, but if you're blood type O, you will contain both anti-A and anti-B type antibodies. Now, people with type AB blood, as I said, don't have any anti-A or anti-B antibodies in their blood plasma. These people are sometimes called universal recipients because theoretically they can receive blood from donors of any blood type because they don't have any of those antibodies to then react with the antigens. People who have type O blood, so they have neither A or B antigens on their red blood cells, are sometimes called universal donors because theoretically they can donate to all of the 4 different blood types, although a type O person can only receive blood from another type O person. So people with type A blood have the A antigen anti-B antibodies. Those with the type B blood have the B antigens and the anti-A antibodies. Type AB blood have the AB antigens and neither antibodies. Type O have neither A or B antigens, but both anti-A and anti-B antibodies. The other type of blood group is the Rh blood group, named so because of the Rh antigen, also called the Rh factor, which is first found in the blood of rhesus monkeys. People whose red blood cells contain Rh antigens are designated Rh positive. Those who don't have these antigens are Rh negative. This is what it means when you say have O positive blood or O negative blood. Now, normally in blood plasma, we don't contain any Rh antibodies. However, if an Rh negative person receives a blood transfusion of Rh positive blood, the immune system of that recipient will start to make Rh antibodies that remain in the blood. If a second transfusion of Rh positive blood is then given at a later date, that clumping or that agglutination will then occur and you can have the destruction of the red blood cells. Rh blood group is really important in pregnancy when you might have a mixing of blood between the mother and the fetus. Not as important in the first pregnancy if the body doesn't have any of those Rh antibodies. Potentially dangerous in the second pregnancy if you do have those Rh antibodies present in that blood plasma. And that brings us to the end of the anatomy and cardiovascular system lecture. Lots to cover with the heart, the blood vessels, and the blood. Next up, we have the physiology of the cardiovascular system, where we will take a bit of a closer look at how all these structures and components work. But thank you very much for listening, and I will see you next time.